What's up guys? In this video you will learn how easy it is to ferment cream cheese in your very own home. To make cream cheese you will need 3 liters of 3.25% whole milk, a quarter teaspoon mesophilic powder culture, liquid rennet, a thermometer, pH paper and cheesecloth. So the first thing you're going to do is pour the milk into a saucepan and slowly heat it up to 62 degrees Celsius on your stove using a thermometer. Heating milk to this temperature is called pasteurization, which can prevent Listeria monocytogenes growth in your cream cheese. Listeria causes people to be infected with a foodborne disease called listeriosis. Listeria monocytogen is a pathogen that can survive a wide range of temperature and pH. It can even multiply in refrigerated conditions. Additionally, the formation of curd during cheese making further concentrates the number of listeria to obtain tenfolds, which can amplify the risk of listeriosis. Consumption of listeria-contaminated food leads to a high case fatality rate among susceptible populations. Okay, so after you have heated your milk, you're going to cool it down to 20-24 degrees Celsius in a cool water bath, and then add 1/4 teaspoon of the powder culture containing mesophilic lactic acid bacteria. Mesophilic lab culture is ideal to be used at home because it has an optimum growth temperature between 27 to 33 degrees Celsius. The lactic acid bacteria in the starter culture are able to convert the lactose in milk into lactic acid. The addition of starter culture can yield a safe and more consistent product. The increase in lactic acid gradually decreases the pH of the milk and initiates acid coagulation by neutralizing the polar K-casein, which is a milk protein. After mixing the milk and mesophilic culture together, add 2 to 3 drops of liquid rennet and stir well. The role of rennet in cheese production is to initiate rennet coagulation. Rennet is a chymosine that contains proteases that act on K-casein. K-casein is important for the colloidal of casein micelles in milk. It has a hydrophobic side that is attached to the micelle and a hydrophilic portion that interacts with the surrounding environment, which allows casein micelles to suspend in milk without aggregation. Rennet can hydrolyze K-casein into para-K-casein and casein micropeptide. The hydrolysis of K-casein polar layer results in non-polar micelles. These micelles are unable to interact with polar water molecules and are forced to aggregate into chains. The micellar chains interact with each other and interlock to form a casein matrix that entraps liquid and water. As surface K-caseins being extensively hydrolyzed by rennet enzyme, the casein micelles aggregate even more and finally reach a stage that they coagulate at any temperature above 18 degrees Celsius. Rennet coagulation happens at high pH of 6.3 to 6.6 and forms the casein matrix that has the ability to expel and separate curd from whey. After mixing the rennet into your milk mixture, cover the pot and allow it to set at room temperature for 14 to 16 hours. After this fermentation period, you should see that a firm curd has been formed with clear whey on top. Carefully pour your entire mixture into some cheesecloth and let it drain for 8 to 12 hours to remove any excess whey. The optimal pH in the final cheese product should be around 4.6, which can be measured using pH paper. And there you have it, your own homemade cream cheese. Hope you enjoyed it.